is a DNA molecule. Now, what if this one DNA molecule turns into 2, 4, 8, 16, 32, and more until it ultimately covers up the entire screen? So let's put it this way. Think of DNA as an instruction manual for building and operating our cell. Every time a cell divides, a whole new instruction manual needs to be built so that we're growing and reproducing as individuals.
Let's talk about what DNA actually is. DNA stands for deoxyribonucleic acid. The information in DNA is stored as a code, which is made up of 4 different chemical bases: adenine, thymine, cytosine, and guanine. And even though there's only 4 different bases, the order of the bases help to identify information that's able to build an organism. So just like the alphabet, even though there's only 26 letters, the way they're structured helps figure out what word or phrase you may be saying. But the DNA bases don't work alone. Adenine binds to thymine, and cytosine binds to guanine with hydrogen bonds. Now, each of these bases are also paired to a sugar molecule and a phosphate molecule. You can think of the structure of DNA like a ladder. The base pairs are the rungs, while the sugar and phosphate molecules form the side pieces. And when you twist the entire structure, it forms what we know as the DNA double helix.
Let's talk about one of its most important qualities: its ability to replicate itself. This is something that scientists like to call DNA replication. So that means a DNA double helix molecule like this one can duplicate itself to make another identical one. Every single second, about 1 million cells die. That's 1 million. That's 2 million. You get the point. Old and broken cells need to go away, so cells dying is a part of the system that helps to clean and sanitize it. But to replace those dying cells, that's where DNA replication comes to the rescue. But how exactly does this happen? Replication has 3 steps.
Step 1. Separation. Let's pretend like both sides of the zipper is 2 separate strands of a DNA double helix. The job of an initiator protein is to unwind a little bit of the DNA, just to get it started. Now a protein, called helicase, attaches to the DNA molecule and breaks apart the hydrogen bonds that keep the bases together. Step 2. Priming. Now as a helicase is doing its work and unzipping the rest of the DNA molecule, another enzyme called primase attaches to each strand and forms a foundation called a primer, which is a short segment of bases that bind to the DNA molecule. Step 3. Building. After the primer is on the one single strand, the DNA polymerase wraps around the strand and attaches new nucleotides to the bases that aren't paired with anything. It uses floating bases to perform something called complementary base pairing. Therefore, we have a new DNA strand built.
So now let's recap the steps: separation, priming, and building. But wait, it doesn't stop here. We can solve incurable diseases and cure them by using gene therapy. And secondly, outside of the medical industry, we can also look at how we can build renewable energy resources out of DNA replication. So the next time you get a really, really bad paper cut and your skin heals from it, just remember, you have DNA replication to thank for it.